A key element of the UK Spine's mission is to support development of drugs which target biological drivers of aging. Toward this goal, the UK Spine's Bridge program has funded the project targeting autophagy for the treatment of osteoarthritis. The principal investigator is Dr. Ghada Alsaleh, postdoctoral researcher in immunology and autophagy with the Simon Group at the Kennedy Institute of Rheumatology at the University of Oxford. Autophagy is the main cellular bulk degradation pathway, and its efficacy decreases with age, enabling an accumulation of damaged macromolecules and organelles which may contribute to age-associated diseases. Transcription factor EB regulates the transcription of lysosomal and autophagic genes, and Simon Lab data confirms that TFEB levels decrease with age in human peripheral blood mononuclear cells. A novel signaling pathway for inducing autophagy involves post-translational donation of spermidine's aminobutyl moiety to a specific lysine on EIF5A known to regulate TFEB. The diagram is from a 2019 Molecular Cell article and depicts relationships between regulation of autophagy via spermidine's influence on TFEB expression. Spermidine itself is not a good candidate for development as a regulatory approved therapeutic as it is too pleiotropic. This work aims to identify alternate targets for modulating autophagy via regulation of TFEB expression, including those in the EIF5A TFEB pathway. The challenge of identifying these new targets is approached in this project via a genome-wide pooled CRISPR screen in conjunction with an osteoarthritic model, selected as osteoarthritis in mice is ameliorated in response to spermidine treatment and resulting increased expression of TFEB. Top hits will be validated with in vitro assays. The single plasmid system Toronto Knockout B3 genome-wide library targeting 18,053 protein-coding genes will be employed to transduce fibroblasts from osteoarthritic patients with 500 single guide RNAs per target to maintain library complexity throughout the screen. Post-antibiotic selection, cells will be pooled for TFEB intracellular staining. The specificity of the staining has been validated using TFEB knockout cells. Stain-identified cells are flow-sorted and deep-sequenced to identify the enriched genes in this population. Following the primary screen, hits will be reconfirmed in a repeat experiment under similar conditions. The data from the screening activities will be fed into Target Hunter bioinformatics platform for annotation, and the top 5 hits will be validated in vitro in chondrocytes and fibroblasts of young, healthy old, and osteoarthritic donors via knockout and subsequent staining for TFEB by flow cytometry or lysed for TFEB EIF5A hypocyanation and LC3 Western blot. Effect on cartilage markers will be assessed by RT-qPCR and Western blot, and cytokine secretion detected by qPCR and ELISA. By identifying novel targets for enhancing autophagy, this work will generate opportunities for new drug discovery efforts focused on these new targets. The specific outputs will support applications for funding to leverage the hits identified in the course of this work in a drug screen to identify promising starting points for further drug discovery activities. This autophagy pathway is likely to be conserved, and results can guide studies with other disease models. Success on this project would not be possible without the collaborations to access the necessary biosamples and conduct the CRISPR-Cas9 screen. This is a very early stage drug discovery effort which benefits from early engagement with downstream partners to streamline experimental design and accelerate translational opportunities. The research group is active in pursuing opportunities to engage with the broader community, including participation in community science festivals and outreach to students at local schools. Drugs designed to mitigate aging would be protective against multiple morbidities via a single intervention. Our current understanding of biological mechanisms driving aging is incomplete. Dr. Alsaleh offers her thoughts on the question, how will this project contribute to a better understanding of the biological mechanisms of aging?
My project focuses on osteoarthritis, which is the most common form of arthritis in the UK. Osteoarthritis is a complex disease with several identified risk factors. However, aging is considered the biggest risk factor for osteoarthritis development. Many evidence point to the impact of age-induced change in the joint tissue in the onset and the development of osteoarthritis. From my own research in immune cells and the promising studies in the literature, We know that the TFEB pathway, a pathway controlling autophagy, the bulk removal of cellular debris, is often dysregulated in age and seems to influence osteoarthritis pathology. My overall aim is to identify the contribution of age-related TFEB pathway to the development and the progression of osteoarthritis, and to use this knowledge to develop new treatment for osteoarthritis. So in this project, I aim to answer several questions. Does TFEB pathway drive aging process in various joint cell type and to promote osteoarthritis? Does targeting TFEB pathway prevent the development of osteoarthritis? And finally, which drug can sustain TFEB expression and thus alert osteoarthritis pathology? As this pathway is more likely to be conserved across different tissue, I hope this research helped to identify drugs that may prove efficient in addressing other age-related disease in addition to osteoarthritis. Overall, we hope understanding biological pathway that change during aging may help us to age healthier in the future.